Thank you, everybody, for coming. The important people are in the room. The less important people are going to miss a true feast of intellectual wealth that our department presents. It is my pleasure to introduce my colleague, Dr. Mark Osborn. He is trained here. He got his PhD in microbiology, immunology, and cancer biology here in 2009, and he joined our pediatric faculty the same year. He has taken a slightly different course in his training, which I like very much. He started with the U.S. Army. He was in the 101st Airborne Division before going to get his bachelor's degree in University of Wisconsin-La Crosse. Mark is the go-to person for anything DNA. If it's DNA, he knows it. He knows what to do with it. He can change it. And he has shown this remarkably in a way of getting everything that actually is on the map, on the menu of gene therapy today, from viral-mediated gene therapy to minicircle plasmid-mediated gene therapy to gene editing on the map. And he has been the leader of the research that led to the first in human gene editing of a disease-causing gene in the context of the human genome. So there are quite a few priorities, that are on the scientific front of our department that are on the shoulders of Dr. Osborn. In addition to that, he's not your regular PhD in the department or in the division. He's someone who goes to the operating room. Sometimes he goes to the morgue with me when we need to collect samples. The patient families love him, and he's got this deep-seated connection between the science he does in the lab and the people we all serve in clinic. Mark?
Thank you very much for that incredibly kind introduction, Dr. Tolar, and it's my pleasure to be here today to talk about gene editing for Fanconi anemia. Importantly, I'm going to use Fanconi anemia, FA, as a model disease as we develop next-generation therapies employing genome editing technologies and stem cell engineering technologies. And so we'll define and establish proof of principle using FA as a model, but the important thing is that these are highly transferable and translatable both to the clinic for many of the diseases treated by hematopoietic or any cell transplant. And so focusing on FA, I'll talk about the biology of this disorder, the current generation treatment, and then our efforts to expand on that to next generation therapies with gene editing and stem cell engineering. So FA is due to a mutation of any one of the FA-associated genes that encode proteins that are involved in the repair of a specific DNA lesion called a DNA cross-link. The inability or the mutation of any of these genes renders the cells unable to resolve these DNA cross-links, and this inability to properly restore and repair DNA results in a greater susceptibility to tumors in these patients. There's multi-organ system involvement, and the most life-threatening manifestation is a progressive exhaustion of the hematopoietic stem cell compartment, resulting in life-threatening bone marrow failure. The FA genes encode proteins that are key mediators, recruiters, and downstream modulators of the DNA repair pathway system. And in response to interstrand crosslinks, a core complex comprised of many Fanconi anemia encoded gene products forms and recruits and mediates the DNA repair process for these interstrand crosslinks. And really the take home with this is that this cross-linked DNA, as opposed to the canonical Watson-Crick base pairing, which is characterized by weak hydrogen bonds, is a very strong covalent bond. And the end result of that is during replication, an interstrand crosslink can stall the replication fork, resulting in stalled DNA replication. In response to that, the FA pathway is mobilized and recruits effectors to resolve this. And its absence or the accumulation of interstrand crosslinks up to as little as 20 can be toxic to the cell. And so the failure to resolve interstrand cross-links results in a failure to replicate DNA. And in the hematopoietic stem cell compartment, the hematopoietic stem cell, which has self-renewing properties, as well as gives rise to all of the progeny and terminal effectors of the blood and immune system, all of which are reliant on replication of this cell population. And its inability to replicate and divide results in a progressive exhaustion of this pool and ultimately bone marrow failure, as opposed to normal people who retain this capacity over their lifespan. And so by about age 7, these patients begin to present with bone marrow failure. And the only treatment for this life-threatening manifestation of the disease is hematopoietic stem cell transplantation. And the procedure for this is that radiation, chemotherapy are given to the recipient to remove the old immune system and bone marrow compartment. Donor cells obtained from an unrelated donor are infused, which then reconstitute the marrow and blood-forming capacity. Work done here, pioneering work done here in the FA clinic, has improved the recovery post-transplant by modifying and improving the conditioning regimen by including fludarabine, which results in improved engraftment at a sooner time. And overall, in event-free transplants, survivability can be quite impressive. However, approximately 2/3 of the patients will have had a prior transfusion, and a percentage of the patients will have had prior opportunistic infections, both of which decrease survivability prior to the transplant. And so while curative and while amazing strides have been made with bone marrow transplantation, there remain significant drawbacks. One is that the conditioning regimen, as well as the immunosuppressive drugs that are given during the engraftment period, leaves the patients prone to life-threatening infections. The infusion of unrelated donor cells has the possibility of initiating graft versus host disease, and so urgent therapies are needed to address these manifestations. And FA really lends itself to the development of both gene therapy, gene editing, and autologous therapies, whereby a patient's hematopoietic stem cells are collected, a functional copy of a Fanconi anemia gene is introduced by a lentiviral gene therapy vehicle. An autologous setting would support decreased conditioning and infusion of the cells, resulting in enhanced engraftment. And an important aside, and one of the reasons why we choose FA to work with, is that corrected cells have a proliferative and survival advantage over uncorrected cells, and so it really lends itself to the development of generation therapies. Currently, gene therapy is being employed in early phase clinical trials for FA, utilizing lentiviral gene therapy vehicles. And so functional copies of whatever FA gene that's defective are provided in a lentiviral gene therapy construct. These integrate into the genome, and the advantage of that is that all progeny cells derived from that will therefore carry that genetic material and have their FA pathway functionally restored. Inherent to how the gene therapy vectors are constructed is that gene expression of this functional copy is often mediated at very high levels in a very sustained manner. And I'll come back to those 2 points because those are important considerations for the advance of gene editing over gene therapy. So while gene therapy has incredible promise, I did mention that the low numbers of hematopoietic progenitors may be somewhat limiting. It becomes a balance between the number of cells available in that particular patient and the low number of cells that are actually needed to affect a therapeutic benefit, because recall that corrected cells have an advantage over uncorrected cells. Current gene therapy vehicles integrate into the genome, and again, their expression is unregulated. And the importance of that is shown in this photomicrograph. So this is just a cell. The condensed regions are closed portions of the genome, and the open regions are shown here. And so this is the areas where gene therapy vehicles are semi-random, and they typically occur in regions of actively transcribed genes. The importance of this is that small genetic changes are sufficient to cause disease. FA is caused by a single base change out of the 3.2 billion base pairs. And so small changes to the DNA code, literally the book of life, can render genes uninterpretable, nonsensical, and non-functional. And so that really provides impetus for our therapies. And these considerations have been highlighted in early gene therapy trials for severe combined immunodeficiency, where an integrating viral construct that mediated high levels of gene expression integrated proximal to a growth controlling gene, and because of these high level gene expression regulatory elements, it dysregulated this gene and drove these cells to a leukemic phenotype. And so in 4 out of the 10 patients, it caused leukemia when the intent was to cure them of severe combined immunodeficiency. And so normal gene expression can be thought of as traffic lights. There's red, yellow, and green that are subject to endogenous and exogenous cues that regulate gene expression in a very controlled manner. Viral gene expression in gene therapy is essentially a green light. It's always on. And so these two factors combine to make very real risks for gene therapy utilizing integrating vectors due to genome disruption and the exogenous gene regulation. However, balanced for that is the benefit of utilizing autologous cells, which would allow for a reduction in the conditioning agent dose and better engraftment. And so we hope to capitalize on that aspect of the autologous therapies and employ gene correction utilizing very site-specific gene editing technologies where we correct the DNA base causative of the mutation in situ. The importance of that is that that's the only alteration to the genome, and it maintains control under the endogenous regulatory elements, and we're employing this as we go forward to establish proof of principle in therapeutically relevant cell populations. And so our ultimate goal is to develop individualized, personalized regenerative medicine therapies that address the entire spectrum of diseases like FA that have both a hematopoietic component as well as other deformities outside the hematopoietic system, whereby we can collect cells, correct them in a very precise manner, select for that event, and reinfuse them back into the patient. And what allows us to do this is the concept of gene editing or precision molecular medicine. And similar to gene therapy, it allows for very stable modifications of the genome. So every cell that's corrected, all the progeny cells are contain this corrected DNA sequence as well. And again, importantly, it's done in situ at the disease-causing stretch of sequence that's mutated, and this maintains gene control under the very tight and localized cues of the endogenous elements, particularly in a DNA damage response pathway whose sustained gene expression may render those cells resistant to apoptosis, while our approach maintains gene control under the endogenous elements. So to accomplish this, we utilize specialized tools that can travel to user-defined, localized sequences by engineering these custom materials that co-localize at a user-defined sequence. It can excise that damaged sequence, and we can insert a user-defined corrected sequence and permanently repair the cell utilizing an endogenous host repair pathway called homologous recombination, which is the exchange of DNA between two highly similar sequences. So that occurs endogenously, but what we can do with genome engineering is introduce an artificial template for DNA repair and induce the cell to repair itself off that. And the frequency of this endogenously is very low. However, by introducing DNA breaks, it can be increased substantially. And so the key to gene editing is creating double-stranded breaks that are very targeted and very precisely regulated. And this is highlighted by many tumor therapies that utilize irradiation, which causes DNA damage and cell death. The accumulation of DNA breaks can also cause toxicity in normal cells. And so by inducing DNA breaks, we want to very precisely control where these occur. And the tool we utilize to accomplish this is the CRISPR-Cas9 system, clustered regularly interspaced palindromic repeats, which is a bacterial adaptive immune system that's been repurposed and re-engineered for mammalian cell use. So in bacteria, phage DNA, which are viral particles which infect bacteria, inject their genome into the cell. The CRISPR-Cas9 system is upregulated. It recognizes these sequences and mediates double-stranded DNA breaks, rendering them non-pathogenic. And it also has the ability to differentiate this sequence from its bacterial genome, and so it can regulate where these DNA breaks occur. And that's what we do in normal cells. So this system has been repurposed and re-engineered for mammalian cell use, and the architecture of it is a two-component system that has a DNA binding component and a nuclease component that actually cleaves the DNA. So the DNA binding component is a short RNA transcript that recognizes a target sequence that is defined by the user. It localizes at that sequence due to simple Watson-Crick base pairing, and then it also has this scaffold that's recognized by the Cas9 nuclease. Once co-localized at the target site, the nuclease wraps itself around the endogenous double-stranded DNA helix, and there's two domains, each responsible for cleaving one of two strands in the DNA helix. This is important because if the endogenous Cas9 nuclease that cleaves both strands of the DNA can, as you recall, I mentioned that homologous recombination is upregulated in response to double-stranded DNA breaks. However, there's also competing error-prone pathways. The easiest way for a cell to repair itself is simply to glue the DNA back together. This is an error-prone process that produces insertions and deletions, and if you remember the Book of Life slide, it shows that small changes can induce very catastrophic and very significant changes. And so there's a competing factor between error-free and error-prone in response to a double-stranded DNA break. When we inactivate one of these domains into a form called a nickase that can only nick one strand of the DNA, it preferentially repairs the cell in an error-free, I lost my pointer, but it repairs the cell in an error-free manner. And so to measure this, we engineered a reporter system that simultaneously measures both error-prone and error-free DNA repair pathways. At top is shown a green fluorescent protein reporter that we can repair, and it turns the cell green only in response to error-free DNA repair. Simultaneously, we can measure error-prone events that turn on a red fluorescent protein. And so we can measure these in cells that were treated with a nuclease that cleaves both strands of DNA or a nickase that only cleaves one strand. And so at bottom left, what is shown is that homologous recombination rates, shown in green, are highest when we utilize a nuclease. However, the mutagenic red fluorescent protein, shown in the red bar, surpasses that, and so it's a competition, and the mutagenic repair pathway is preferred. However, when we utilize single-stranded DNA cutting enzymes, the overall rates of homologous recombination or error-free gene repair is decreased in comparison to the nuclease. However, the error-prone pathway is essentially at baseline, and so when we express this as a ratio of error-free to error-prone, the single-stranded cleaving enzymes preferentially utilize error-free DNA repair pathways, and that served as our platform for generating a custom-designed reagent for use in a Fanconi Anemia C patient with a very common mutation. We obtained skin biopsies from this patient, generated a fibroblast cell line, created the custom gene editing tools, introduced them into the cell in the context of an artificial repair template that would allow for permanent correction utilizing that template. And so the overall schema is shown at top left. That's the Fanconi Anemia C locus. The black bars underneath are our artificial template that share homology to the endogenous locus. The mutation site is the red arrow, and so in untreated cells, the mutated base shown in the DNA chromatogram stays T. However, when we introduce the gene editing materials, we're able to revert that base back to wild-type status, which is the A. And we did the comparison in Fanconi anemia with nuclease, or enzyme that cuts both strands of DNA, and the nickase that cuts a single strand. And similar to the previous slide data, we show that the nickase preferentially utilizes homologous recombination. We were able to achieve high efficiency gene correction in a patient-derived population of cells. This particular mutation results in exon skipping of exon 4 in the Fanconi anemia C gene, which prevents its participation in the core complex, and a failure to repair DNA. And so to show that we were able to revert the base at the DNA level, we also looked for restoration of exon 4, and at the bottom right is where we show in the nuclease and nickase treated cells, we were able to re-express this missing exon and render the cell functionally able to repair DNA at the DNA, RNA, and protein levels. And so we generated this custom tool, this molecular scalpel, specific for this Fanconi anemia C mutation. It's approximately 15 base pairs away from the base that causes the disease. And just because we design it for that spot, and the rationale was similar to if we were to open a phone book, turn to the Fanconi anemia C page, this is the base we want to change, and put our finger on it, and that's what we're going to target. Sequences within the genome can have homology to other sequences in the genome. And since this is a DNA binding event, the possibility is there for off-target effects, which would be similar to turning to the Smith or Johnson page of the phone book. They're highly similar at face value, but individually are highly unique. And so what we did for the first time with this class of reagents was took an unbiased view at the genome level, as well as utilize computer modeling to show that this was an exquisitely specific reagent that we had generated. And so what the table at top shows is the Fanconi anemia C gene sequence that we've targeted. Below that are computer modeled sequences identified that scan the genome and look for matches to the intended target site. And we did not observe any activity of this reagent at these genes, and we expanded that again to the genome level and did not exhibit activity anywhere other than the intended Fanconi anemia C target. And so in conjunction with utilizing the nickase, which cleaves a single strand of DNA and efficiently repairs the DNA, resulting in correction of the DNA and RNA and protein levels, we also showed exquisite specificity, which will be required for clinical application. What we had done then is utilized a cell population that is unable to repair certain lesions of DNA, induced DNA breaks, and asked it to repair itself, and we're able to do that very efficiently and very precisely. And the cells we chose for this were fibroblasts, and so this will serve as proof of principle as an expansion to more therapeutically relevant cell types, in particular hematopoietic stem cells. Recall that these cells are limiting in terms of number. Homologous recombination in hematopoietic stem cells only last year was first described at a Nature level publication. And so there's certainly hurdles to this approach, and the way we intend to address those hurdles is through a multi-pronged approach. One approach is to expand the limiting number of stem cells obtained from a Fanconi anemia patient to sufficient numbers where we can intervene, correct the cell, and then deliver them back to the patient. And what we're doing is, in conjunction with ongoing work done in the university, in our division, is utilize a molecule called SR1, which preferentially expands hematopoietic stem cells and, importantly, maintains them in their pluripotent state so that they can continue to engraft. Clinical studies performed here show that expanded cells maintain activity and also increase efficiency in regards to the number of cells expanded resulted in a quicker restoration of neutrophil counts, which is shown at bottom. And another approach that we're taking that will also take place in parallel, intended to both address the hematopoietic complications of the disease, as well as the extrahepatic complications of the disease, is to utilize stem cell engineering. The classical differentiation process is from a primitive cell type to more defined cell types that are the terminal effectors. For stem cell engineering, what we can do is take an adult cell population, introduce factors that induce them to become pluripotent, and adopt the ability to differentiate into many tissues. One aspect of this then is to correct cells, as we've done with the fibroblasts, induce them into cells that can then turn into many other cell types. And intense research is going into defining the factors and pathways that we can modulate and recapitulate in vitro so that we can form definitive type hematopoietic stem cells capable of engrafting. I also mentioned that hematopoiesis considerations are but one aspect of FA, and so what we're also doing is utilizing stem cell technologies to differentiate into many other tissues as part of a regenerative medicine approach to treat the entire spectrum of the disease. And so what we term these are next generation therapies that are progressing at a pace that we hope will be applicable to the current generation of patients. The individualized process is that each individual patient, we can generate a gene editing reagent in a matter of days and very thoroughly and relatively quickly correct whatever disease causing mutation that they have at the DNA level. We hope to then feed this into a standardized pipeline for hematopoietic stem cell expansion and next generation stem cell engineering. And to say standardized is in no way meant to trivialize the enormous amount of work that is and will go into this. But an individual patient utilizing refined and optimized and standardized hematopoietic cell expansion and supportive culture techniques, as well as regenerative medicine, will allow us to individualize the therapies and enhance their ability to engraft and do so as a number of tissues. The importance of this is that we've established proof of principle for Fanconi anemia, that a DNA deficient cell can repair itself if we employ single-strand nicking enzymes, and can do so very specifically and very robustly. And importantly, it serves as a stepping stone not only to other bone marrow failure syndromes seen at the bone marrow transplant clinic here, but many of the disorders that are seen here and treated by hematopoietic cell transplantation. And so what gene editing and stem cell expansion, stem cell engineering, and differentiation technologies are really acting as force multipliers. The permanent correction can be fed into therapeutic pipelines, and it's really a synergistic approach utilizing multiple disciplines, all coming to a singular focused angle of attack for translational application for Fanconi anemia and many other disorders. And importantly, the system we're using, I talked and focused on permanent gene correction today. Cells can also be engineered. We can inactivate deleterious genes, so genes that are responsible for causing graft versus host disease. We can inactivate those as part of an enhanced cellular engineering approach to increase engraftment. The CRISPR-Cas9 system is incredibly flexible. It allows us to do in vitro disease modeling, drug modeling, and gene discovery. It also allows us to induce and employ less permanent alterations, whereby we don't have to either change a sequence or inactivate or delete a sequence. We can actually modulate the expression of genes in a very tuned and tightly regulated manner. And so we can induce cells to adopt different properties without changing the genome. And so these are all really progressing in a synergistic approach. And really wouldn't have been able without the group I'm privileged to be able to collaborate, co-work, and be mentored by. Particularly of aid has been the Children's Cancer Research Fund and the Kids First Fund, which has been a phenomenal partner in cooperating and aiding us as we translate these therapies into a translational application for human use. I'm going to stop there, and I'm happy to take any questions.
your boss. So are there areas of the genome that are more available to this CRISPR-Cas-based 9 system than others that
and that's a fantastic question, because I glossed over that a little bit. So the system we used is derived from the strep pyogeny strain of bacteria. That recognizes, it has to have a 3-base recognition sequence in the context of whatever gene we're targeting. There are other isoforms or other bacterial species that have this system, and that's where the diversity comes. And so as we employ more and more of these, the flexibility increases where we can target multiple spots in the genome. And importantly, by utilizing differential ones simultaneously, we can start to do 2 things at once without cross-reactivity. And so we can utilize the properties of each of the individual enzymes to our advantage, both to expand specificity and our target coverage, as well as do multiple things simultaneously.
Do you have any concerns for, are you planning, when you're doing this for each individual patient, to look at the specificity at the genome level, since you'll be doing different sequences?
Yep, so that's a really good question. I don't think that the field is in agreement yet. Some of the leaders for the field were in Boston recently at a meeting I was at, and genome sequencing actually wasn't advocated at that stage, that the coverage isn't enough in a big enough population of cells. And so what we do is these more technically advanced methodologies whereby we map differential DNA break sites utilizing deep sequencing, and so we achieve coverage at that level. But whole genome sequencing probably isn't cost effective, and the resolution probably isn't at the state where it would be applicable for many patients. But I think that eventually the field will move there as so the important thing is some of the genome scans we do feed back then into the engineering of the enzyme to make it more specific. And so the hope is, while we're at the top of the funnel, is to refine the enzyme so that we can then look at a very specific number of sites within the genome as part of a quality control, quality assurance, to say this is a safe cell to transplant back into a person. But whole genome sequencing doesn't accomplish that at present.
So I think the application of this really makes sense when we're talking about hematopoietic stem cells, because they have, as you said, they'll have the corrected cell will have a proliferative advantage. The cells anyway are, all the stem cells are in a proliferative state. But when it comes to iPS cells, I mean, obviously, especially for FA, we want to correct this stem cell, the hematopoietic stem cell, but the other goal is to correct the rest of the cells in the body, particularly to decrease the risk of malignancy. So my question is, if you correct the cell using iPS technology and have them differentiate and proliferate into different cells, how can you then make it that they have a proliferative advantage, let's say in skin, to reduce skin cancer, or head and neck, where the FA patients have cancer, when most of those cells are quiet?
That's an excellent question. I think that that's certainly one of the key challenges to stem cell engineering, is what is the threshold to achieve. I can't envision where we would irradiate the skin to cause proliferation. What I could envision is ex vivo platform organogenesis, or skin manufacturing for grafting or placement into an organ. And then I think it becomes a question of the biology. In FA, will they eventually repopulate the organ like they would in the hematopoietic system? And I don't know whether it would. In terms of say the oral cavity, that's an interesting component, because what we're also doing is looking at other factors that may be drivers of malignancy, the oral microbiome or things like that. So the advantage of this is we can correct human cells. It's also derived from a bacteria, and so antibacterial properties can be employed for this. And so we could theoretically engineer the microbiome into a more favorable state. And I think that as these systems progress, ex vivo organogenesis or defining the gut or oral microbiomes, then we can begin to envision strategies for modulating those in conjunction with gene editing. But I think that, as you and I discussed, that the hematopoietic system really lends itself to this process. But I think that the stem cell engineering field is certainly being mobilized to address the extra hematopoietic components of this and many other disorders.
So I have similar interests to Margie. So you can fix the hematopoietic system, but the rest of the problems are still there. Is there any movement to doing in vivo gene therapy with this system, or where you can actually, I don't know, inject CRISPR-Cas that can go and correct in vivo problems with the other cells as opposed to needing to use IPS?
Absolutely, and at least two approaches are being adopted for that. One is direct gene correction. So one of the most accessible organs for this that's been in animals has been the liver, where this material can be injected, particularly for hemophilia, where very low levels of correction are required. And by targeting the liver, we can use that as a depot organ to achieve systemic benefit. But I think that that's, again, back to Margie's comment, that is certainly a potential hurdle. But in vivo application, with the skin accessibility, that's certainly a benefit of this. And there are in vivo gene editing and as well as targeted, as well as knocking in genes into, say, the albumin locus, which makes very high protein levels. And so some of the enzymopathies for lysosomal storage disease, knocking in a gene into that region where we know we'll get very high level, and as opposed to gene therapy where we don't know where it's going to go, it'll go to one spot and infuse enzyme into the circulation. Those are the two primary goals of liver-directed gene therapy at present that have been accomplished in animals.
I knew it's going to be fantastic, but now really, blue sky thinking, related to, say, your work on the T-CARs or, having a cell that's not present in nature, not present in the human body, and can target it somewhere. Can you combine it, or can you think of how to combine that technology, which is already in trials, with a CRISPR-based technology that would regulate RNA level, that you would have a cell that would actually have a whole complement, whole menu of functions that can be put in action and then taken away?
Absolutely, and I think that Dr. Neglia's question kind of feeds into that utilizing these different orders of CRISPR-Cas9 system that we can engineer to essentially act as a switch or a stepwise progression where one gene controls the regulation of the next and the next and the next. And so we can fine-tune these things, and I think we can do it by engineering a cell that we can deliver a drug that would induce the cascade or turn something on for a little while and then remove the drug and turn it off. And so I think that being able to fine-tune these and being able to do it without even taking a step back from the genome level and just doing it at the RNA level is really going to be an exciting avenue of this, because then we can put these things in and control them, and if there are deleterious events, then it's at the RNA level and we can control that. And so I think that this very tightly regulated and ability to control these cascades will really push the forefront of what we can do with cellular therapies and be able to do it in an in vivo setting. And so take everything out and engineer the cell and then put it in, but maintain the ability to regulate the different cascade through oral drugs or some intervention where we can control very tightly and very precisely not only one gene, but a cascade of genes.
one of the complications is the hepatocellular carcinoma, and I assume that there's mouse models for the Fanconis that you could utilize your portal approach to first.
Absolutely, and even malignancies in general, we've considered translocation events that are causative of malignancies. Can we introduce these in vivo and disrupt the translocation event? So rather than targeting normal sequences, we can target translocations or specified sequences that we know are causative of malignancies. And so from an accessibility standpoint, the liver does lend itself. And utilizing Dr. Toller's example, for a malignancy, we could then consider modulating the apoptotic cascade or something along those lines where we can increase efficacy by either targeting the gene or the process.
Well, thank you, everybody. Remember one thing. You have a internationally recognized expert on this here in this department. It's sometimes difficult, to realize, how close are you to excellence. So if you have a basic science project, I didn't ask your permission, or clinical science project that you think may benefit from this top-notch technology, talk to Mark. He's a nice guy. Thank you, everyone.
Thank you.